Hey everyone, let's understand cellular aging. The aging is a universal biological process. At the cellular level, aging means a progressive decline in the replicative and functional capacity of the cell to divide, function, and survive. It occurs due to cumulative genetic and metabolic damages over time. Let's look at the major mechanisms that explains why cells age. These are the 6 mechanisms by which the cells undergo aging. We'll discuss them one by one, starting with the DNA damage, which can also be said as genomic instability. Throughout life, our cells are exposed to DNA damaging agents like UV light, toxins, which are the exogenous agents, and reactive oxygen species, which are the endogenous agents. While repair mechanisms exist, over time errors accumulate. This genomic instability is a fundamental cause of cellular aging. Next, we have cellular senescence, which is replicative senescence. So, normal cells have a limited number of divisions known as Hayflick limit. With each division, telomeres, which are the protective caps at the end of the chromosome. If this is the chromosome, here's telomere is located, and with each cycle these are shortened. Critically, short telomeres are sensed as DNA damage. This activates tumor suppressors. The most common ones are P53 and RB, forcing the cells into permanent growth arrest, a state called senescence. Interestingly, senescent cells remain metabolically active and secrete inflammatory mediators, growth factors, proteases, which are together known as SASP, that is senescence associated secretory phenotype. Next, there is defective protein homeostasis. The cells maintain protein quality by chaperones, the proteasomes, and autophagy lysosome system. With aging, these systems decline. Misfolded and damaged proteins accumulate, leading to cellular dysfunction and death. Then we have deregulated nutrient sensing. So aging is also linked to altered nutrient sensing pathways, especially insulin and insulin-like growth factor 1 signaling and the mTOR pathway. Increased signaling here promotes aging, while reduced signaling, as seen in caloric restriction, expands the lifespan. Caloric restriction activates sirtuins, which improve DNA repair, mitochondrial function, and stress resistance. Then, mitochondrial dysfunction. So, mitochondria of aged cells show reduced oxidative phosphorylation and increased reactive oxygen species, and accumulation of mitochondrial DNA damage. These all lead to amplification of cellular injury, and this creates a vicious cycle. Then we have epigenetic changes. Aging involves alteration in DNA methylation, histone modification, and chromatin remodeling. These epigenetic changes shift gene expression patterns and reduce the regenerative potential of the cells. Under the microscope, aged cells show irregular nuclei, cytoplasmic vacuoles, lipofuscin, which is the wear and tear pigment, and abnormal mitochondria, that is, their size and shape changes, and also the number and efficiency of the mitochondria is reduced. Now, these mechanisms explain why aging contributes to degenerative diseases like Alzheimer's, Parkinson's, atherosclerosis, etc. They also intersect with the cancer biology, where senescence prevents tumor growth but accumulated mutations may fuel the malignancy. The understanding of these pathways is key to anti-aging research and regenerative medicine. We can modulate aging by certain things like caloric restriction, as we discussed how it helps. Then there is medicines like rapamycin, metformin, which mimics this caloric restriction effects. Then there is lifestyle modification which could reduce the oxidative stress and improve DNA repair capacity. So in short, cellular aging is a multifactorial process. DNA damage, telomerase attrition, cellular senescence, protein misfolding, mitochondrial dysfunction, and nutrient sensing and epigenetic changes. These all combine to reduce cell function and survival. Ultimately, aging reflects the balance between cumulative damage and body's repair mechanisms. Thank you for your time.